I'm oral historian Mike Chappell. Today, June 5th, 2011, I'm interviewing Dr. Samuel Refetoff for the Endocrine Society at its annual meeting being held this year at the Boston Convention and Exhibition Center. Dr. Refetoff, please tell me where your parents were born and what kind of education they had.
Both my parents were born in Bulgaria. My father had a university education in business, and my mother was didn't finish high school, but she left a year before graduation and became a seamstress. But after I was born, my mother didn't need to work anymore.
What did your father do?
My father was a banker. He worked for the Franco-Belgian Bank until the bank was closed during the war, as it was owned by an enemy country, Belgium.
And what was life like for you and your family in Bulgaria during the war and its immediate aftermath?
Well, during the war, we have some deprivation in terms of food, and we had some bombardments by the Allies because Bulgaria was an ally to Germany. But things did not get difficult for my family until after the war, during communism, where families from individuals that are well-to-do were accused of amassing money by exploiting the proletariats. So, in fact, this is where my family left, about 4 years after the end of the war.
And where did you go to?
So, because my father worked for the Belgians, he thought that going to Belgium, we all left as refugees, that he'll find a job. But in fact, it was very difficult for refugees to be employed, even if he did work for the Belgians. As a consequence, we had to manage with some money that my father had saved in European banks, and eventually immigrated to Canada.
What kind of student were you, and what were your favorite subjects?
As any child of refugees who had to work hard to make a living, we had no choice but do well at school. Otherwise, the guilt was quite strong if we didn't. And I think that has been a motivation for me and many other children in my situation. So I did quite well at school, both in high school and later in college. I was not a top student in medical school. I think by that time, the guilt element kind of got diluted.
What about activities, extracurricular activities, sports and things like that?
Yeah, as in high school I played volleyball, but later on in college I got involved in fencing and became quite good at fencing. I almost made it to the Olympics representing Canada, and I was also the captain of the fencing team at the University of Montreal. One of the reasons I think I was admitted to McGill Medical School is because they hoped that I may join the fencing team, because University of Montreal was leading in Canada and McGill wanted to move in that direction. They were really disappointed when I decided that the medical school was quite difficult and continuing fencing was not the right thing to do.
When did you first, and why did you first commit to a career in science and medicine?
Well, that came in steps. As a child, I was interested in biology. I was particularly interested in insects. As a matter of fact, I have continued to collect insects even after medical school. I have quite a good collection of butterflies. And this brought me to biology. I studied biochemistry because I got interested in chemistry and wanted to continue in that field, getting a PhD. I then became graduate student with Charles Philippe Leblond, but didn't like it for various reasons. I think the atmosphere in the lab at the time was a little bit difficult. So being at McGill, I applied to medical school. As a medical student, I got interested more in the practice of medicine. I became very fond of Albert Schweitzer and thought to have a career in tropical medicine. And for various reasons, this didn't pan out. And I, through a pure accident of encountering a family with a difficult endocrine problem related to the thyroid, which was the subject of my work with Leblond, I returned to the thyroid and back to research.
How did you decide to do your internship at Notre Dame Hospital?
I came to McGill from a French-speaking university. I didn't speak English and had some difficulties at the beginning of in my first year of medical school. There was I was also at that time quite active in trying to reduce the animosity between the French and English-speaking individuals, and I thought that with two other friends of mine, one Greek and one a Polish Jew, that we should attempt and break that separation by going into French-speaking hospital to do our internship, which was not, we are the first one that have done that. Which was not, turned out not to be very easy because there was a lot of resistance from both sides. But once this was done by us, then in subsequent years, this continued. English-speaking students from McGill went to French-speaking hospitals and vice versa. And now that is pretty well the custom in Montreal.
How did you come to begin your residency at the Hospital of the Good Samaritan in Los Angeles?
After I finished medical school, I was accepted with my interest in tropical medicine to study at the London School of Tropical Medicine under a scholarship given by the Canadian government. Only months before I was due to go to London, I received a letter saying that the Canadian Parliament debated that there was no really problem in Canada with tropical diseases, and therefore they didn't see what was the value of supporting studies in this area. So I found myself without without my plans, basically had to be changed. I didn't apply for residency, so I looked through the medical journals and found an ad in the New England Journal of Medicine that they had a vacancy at the Good Samaritan Hospital. I think a resident became sick, developed leukemia, and had to drop from the program. And so I was accepted in the program and moved to LA.
Now, what kind of physician scientist were you thinking about for a career?
So at that point, at that breaking point, I was still kind of disappointed that I could not pursue my interest in going to tropical medicine, but had to compromise. And it is at that point that I got involved with a family with an unusual thyroid presentation, which brought me back to my earlier studies with Leblon on the thyroid, and kind of developed and gradually developed interest in research.
What was the state of the art of thyroidology at that time?
I think, well, at that time, we are talking early 60s, late 60s, there was no specific method to measure thyroid hormone. Thyroid hormone was measured indirectly as a protein-bound iodine. Basically, the amount of iodine in protein in the serum was quantitated as possibly representing thyroid hormone. And indeed, in most instances, it did, but there were others where the iodine was contained in some other compounds and where the results were false. It is only in the subsequent years with the development of radioimmunoassay, but first by development of the protein binding assay by Murphy and Patty from, actually both of them from Montreal, a more specific method for measuring thyroxine developed. And it took many years, later 60s, when Kenneth Sterling first published the method to measure triiodothyronine.
Would you speak to the medical or scientific stature of Dr. Loren Dewind?
Dr. Dewind, who incidentally graduated from University of Chicago, where I was to go, was a practicing endocrinologist. I should say that, in my opinion, I may be biased, he was a very devoted and excellent endocrinologist. He had no scientific background. I don't believe he had a single publication at the time. But he was astute enough to recognize the unusual presentation in the family that I ended by studying, and he encouraged me to do so. I was a resident at the time, first-year resident. He helped me to obtain a small grant from the Hospital of the Good Samaritan, which provided enough money to run tests on members of the family. And this is what started the study of the first family with resistance to thyroid hormone.
And did you see your career then has it made a major shift?
I think the shift developed later. I think as I continued studying the family and found it challenging step by step, an interest developed in continuing and in pursuing a career in research.
How did you come to be an assistant in medicine at Peter Bent Brigham and a fellow at Harvard?
Yeah. So because my plans to go to London had to be canceled, and I obtained just a temporary position at the Hospital of the Good Samaritan, I immediately made application for the subsequent years. Although after 1 year, Good Samaritan Hospital wanted to keep me for a second year. By that time, I already secured the position in Boston. Not immediately at the Brigham, but first at the Lay Clinic. So I did another year at the Lay Clinic, following which I joined the Brigham as a fellow in endocrinology under Herb Selenko, who was the thyroidologist at the Brigham, and he became my mentor mainly because I developed that interest in the thyroid.
Now, in an earlier interview that you did for the Endocrine Society with Dr. Adolf Friedman, you sketched out the emergence of your interest in genetics and endocrinology, and you also talked about John Stanberry and Leslie DeGroot. That's on the record already. But I would like you to talk a little bit about the thyroid clinic and the ethos that was going on there, and maybe something about the scientific backgrounds of Dr. Stanberry and Dr. DeGroot.
I was never at the Massachusetts General Hospital where the thyroid clinic took place. I got to know Dr. Stanberry and DeGroot because Dr. Dewind and myself had a little bit difficulty convincing people that we had unusual cases and some difficulty in publishing our results. So at the lay clinic, I was, I think Dr. Elmer Bartels at lay clinic suggested that I go to Mass General and talk to this gentleman. Actually to Dr. Stanberry, and this is where I met Dr. DeGroot, who undertook to send me back to LA to do some additional studies, and eventually resulted in that first publication with the authors being myself and Dr. Dewind and Dr. DeGroot. Both Dr. Stanberry and DeGroot have very high standing in the scientific community, particularly in the thyroid. Dr. Stanberry was the mentor of DeGroot, and I think if it were not for the decision, or the inability to pursue a plan to open a medical school at MIT, both DeGroot and Stanberry would have been still there, and probably myself. Stanberry moved to MIT together with DeGroot. I had hoped to join them in the medical school, and when those plans collapsed, DeGroot left for Chicago and eventually recruited me there.
What did he offer you?
Well, he offered me to be assistant professor, which I didn't expect to become at the Brigham probably for several years.
What were your initial responsibilities there?
Well, like any young faculty at that time, we shared, and probably still now, our time between clinical work and laboratory. The clinical work was probably about 50% of the time. We had to attend general medicine probably 3 months out of the time, and we had a separate endocrinology ward, which now is non-existent in any hospital that I know. And the laboratory work was done in the rest of the time. Now, the universities at the time did not pressure faculty for income. In other terms, there was no direct accountability as to how much revenue there was from patients. So young faculty had reasonably protected time to do their research. Also, research didn't cost that much. We made our own reagents. We didn't have to buy them. So with very little money, we could undertake work. It took time, but certainly could be done quite cheaply. So there was not big pressure to obtain grants to support, certainly to support our salary and so forth, which is now the problem with young people, where they don't have only to secure monies for the reagents and the expenses they have in the lab, but also for the part of the salary they obtain to secure a protected time to do their research.
Now, among your scientific contributions, there's been the identification of 3 separate syndromes: resistance to TH, resistance to TSH, and the defect in thyroid hormone metabolism. Does that have a name for it, that last one, as a syndrome?
Yeah, it's defect in selenoprotein synthesis, because this is probably exactly what it is, because the mutation involves a protein which is absolutely required for the synthesis of selenoproteins.
So those 3 syndromes, in what way does your research on these syndromes typify the approach you've taken in your laboratory?
Well, going back, I would say it became with my gradual involvement in the thyroid. The first family that got me into research had a genetic defect, so that introduced me into to genetics combined with thyroid. I think my exposure with John Stamber and Leslie DeGroot, John especially, who has by that time identified several errors of thyroid hormone synthesis, had something to do with my continuing interest. At a certain point, I took a sabbatical in Brussels in 1975, where I was really introduced by Vassar to serious genetics, and so that took off. Then my career took off, combining thyroid and genetics. The methodology, I would say, the way it evolved with modern development in research is identification of a new syndrome or a new genetic defect, search for the gene responsible for the defect, then producing a mouse model that will replicate the defect, because there is limitation in how much one can do with humans, especially the provision of tissues that are not accessible under normal circumstances. And then studying in more detail the disease in the mouse, which then allows us to go back to the human and try to apply various forms of treatments. For many years, the treatment for thyroid diseases was simple, namely giving thyroid hormone, because most of the conditions we identified or were identified were caused by a deficiency in thyroid hormone. So as we learn more, we are now learning about defects where thyroid hormone, although the defect is absence or deficiency in thyroid hormone in tissues, this cannot be corrected by giving thyroid hormone, because the defect is in the entry of thyroid hormone into the cell. So we have now to be a little bit more creative and to develop treatments that are slightly different, which will bypass the use of thyroid hormone and using compounds that mimic the effect of thyroid hormone but are not thyroid hormone. So that closes this circle from the identification of a condition in the human passing to an animal, which allows in-depth studies back to the human to try to remediate it and to correct it or cure it.
Would you bring your thyroid hormone resistance work up to date from Boston, tracing it through your early days in Chicago and then up to the present day?
So my work on thyroid hormone resistance began in Los Angeles in the latter part of 1964 with the identification of a family with 3 children that had high thyroid hormone levels but did not manifest the expected symptoms and signs that will result from too much hormone, suggesting some form of abnormal hormonal resistance. The initial work went to proving that the hormone was an authentical hormone, which was not easy at the time, where there was no direct test to measure the hormone, which I just mentioned before, and involved a rather more sophisticated biochemistry work, including the use of snake venom to prove that it's an L-amino acid and so forth. So with that, there was a long term of clinical studies, and during which these children were given various forms of thyroid hormone and some of the even analogs of thyroid hormone in studies that took us through the 72. 2 long visits, one in Boston when I was here of 3 months, and of the family of the affected 3 children, and one that followed in Chicago that lasted almost 6 months. The children stayed in Chicago. We provided them schooling and all the rest, which wrapped up the study, and I think we had a case that was convincing that these children had a problem with the recognition of thyroid hormone, namely the receptor. And then there is a hiatus until 1989 because the receptor was unknown. And so in 1989, 2 laboratories, one of Björn Wenström in Sweden and one here in the United States, Evans, identified the 2 thyroid hormone receptor called alpha and beta. One was in humans and the other in chicken. So with the identification of the thyroid hormone receptor, there was a tool now to find whether any of those individuals had a defect in the receptor. By that time, there were at least 15 families already recognized to have such a defect, so we were not the only one that identified a mutation in the thyroid hormone receptor gene. The first recognition that there may be a defect in the thyroid hormone receptor came from work at NIH by Stephen Usala, who was working then in the laboratory of Bruce Weintraub. They did what is called linkage analysis and showed that there was that the affected individual shared an allele in common which was not present in the unaffected individuals of the same family. Now, it is not that we sat idle during that period of time. We were doing the same work. However, and I was in communication with Steve Usala, with whom we became good friends, and we got contrary results. And to be nice, I suggested that he may probably not publish it because there may be an error. It turned out that the error was ours. We mixed the sample of the grandmother and grandfather, and the grandmother was mislabeled as the grandfather and so forth, and so we lost the linkage, which requires that the allele co-segregate together with the phenotype, with the abnormality. We eventually found that this was the case by getting new samples, and that was also complicated because the grandmother in the meantime died, but we dug some of her cells that I had frozen, some skin fibroblasts, and we did that. However, in the next step, really finding the mutation, we kind of made it a little bit ahead of Bruce Weintraub by about a year, or maybe a little bit less than a year. They're published in different calendar years, but probably the difference was only 8 months. And from then on, more and more mutations were identified. So that opened the field into understanding how those mutations caused resistance to thyroid hormone and why it manifested in a dominant fashion where the individual had could synthesize both normal and abnormal receptor. And then the concept of dominant negative effect, which means that the mutant receptor interferes with the function of the normal receptor, arose, and I think opened the field into understanding some aspect of how nuclear receptors work. Now, as work progressed, we could go back to the original family where the inheritance was recessive to find that they didn't have dominant negative effect because instead of having a mutant receptor, they just had deletion of the entire receptor. So there was, when only one was deleted, the other was normal, it didn't give any condition, but when both were dropped off, there was. And in fact, out of probably now more than 3,000 cases, this original family remains the only one that had the deletion of the entire gene. However, and I'm talking more about it because it's the subject of my interest, as many groups got involved in work with the thyroid hormone resistance, it became apparent that there is a subgroup of individuals representing 15% of people that have the clinical phenotype that have no mutations in the receptors. And that has been proven again by co-segregation analysis, proving that it's not that the mutation was missed, but it's impossible that this could be caused by mutation in the receptor. So this is where we stand now. Several groups, including mine, are working hard to find out what is the cause for this thyroid hormone resistance. In our case, we have identified 27 families already. To have the clinical phenotype that is undistinguishable clinically from those with mutation in the receptor, but they don't have mutation in the receptor. There are various theories. Naturally, good candidates are proteins that are involved in the complex that involves the receptor, and through that mediating the gene transcription, but there is no exact cause yet known.
How is this current work being supported?
Well, I've been, I would use the term lucky because there is some degree of luck. Naturally, luck is not sufficient to be funded, but looking at other groups that have done good work and have lost funding, I should include luck in that fact that I've been continually funded by NIH, only a single grant. I'm in my 42nd year and heard a week ago that I got 5 more.
What is resistance to the action of TSH?
It's similar to thyroid hormone resistance, where the thyroid hormone level is high, but the effect is reduced. All those syndromes of resistance, they are partial, they are not total. So they are reduced sensitivity, should be the proper term, but resistance have become the preferred one. Is that these individuals need higher TSH to function normally. In other terms, they have resistance to the normal amounts of TSH. And again, those were identified. The first case, you want to hear about it?
When was resistance to thyrotropin first recognized as a clinical entity?
A young pediatrician by the name of Michael Gottschalk at the University of Loyola just joined the faculty and inherited a number of patients from his predecessor. Among them was a girl who was born with high TSH, thyrotropin, and was put on thyroid hormone. There were 2 other sisters that had the same situation. what he was astute enough to recognize is that firstborn that had also high TSH was not treated with thyroid hormone for 3 years until the second was born, and yet she developed normally. So he rightfully concluded that the TSH was high, but there was no need for thyroid hormone because the girl developed normally. So he contacted me, I reviewed the cases, and we decided under the circumstance to stop thyroid hormone. And in fact, all of them had normal thyroid hormone level but high TSH, suggesting that they really could compensate by having high TSH. We looked at the possibility that TSH had an abnormality and didn't. And so we moved on to the receptor and found the first mutation that reduced the affinity for TSH. Now I should say that my colleague and friend Vassar several years earlier found mutations in the TSH receptor that actually activated it and caused the opposite condition, hyperthyroidism. But those are all somatic mutation in thyroid nodules. At the time, we recognized that. He phoned me and he said that he thought he has a germline mutation now that activated the thyroid hormone. And so we got together and said we'll publish it together. And in fact, that's what happened. Peter Kopp, who was at the time in Switzerland, identified a child that was born with hyperthyroidism and have gain-of-function mutation, and ours, and they were both published in the New England Journal of Medicine, the gain-of-function, loss-of-function mutation, which is how things should be done.
I'd like to ask you a little bit about the SBP2 gene. What are selenoproteins?
Selenoproteins are proteins that contain an unusual amino acid, which is selenocysteine, in one position or several positions in the amino acid chain, and that qualifies them to be selenoproteins. There are about 24 and probably more known selenoproteins that have various function. In some of them the function is unknown, but there is a separate interest in that field of selenoproteins.
What is the function or the functions of the selenocysteine insertion sequence binding protein 2 gene?
Right. So to become a selenoprotein, one has to insert a selenocysteine in a position in the coding sequence of the RNA. Now the code for insertion of selenocysteine is a stop codon normally. It's a UGA, which is a stop. So in some instances, that stop is recoded so that the protein synthesis is not stopped at that stop codon, but the selenocysteine is inserted, and then that allows for other amino acids to be added in the sequence until a true stop occurs. Now, what determines that a stop will be recoded to put a selenocysteine is a signal that is contained in the non-coding sequence of the RNA. And to recognize that signal, the selenoprotein, the SBP2 binds to that signal and determines in the synthesis of the protein that next stop be recorded and a selenocysteine be put and synthesis should continue.
What brought SBP2 gene defects to your attention?
As with all other instances, it starts by an observation of a defect in the thyroid. In this instance, the first family were children from Saudi Arabia, who were brought to attention because one of them had some growth problem. The testing showed no growth hormone abnormality, but instead a very high level of thyroxine T4, but not the active hormone T3, which then suggested that they have a defect in the formation of the active hormone, and maybe because of that, the growth defect. There are three enzymes that activate the precursor of thyroid hormone, T4 into T3, and those are called deiodinases. So it was only logical to suspect the defect will be in one of the deiodinases, and we looked at all 3 deiodinases and found no defect. Deiodinases are selenoproteins. So the next step was to do something back clinically to see what could be another possibility. So we took skin biopsies from the individual, grew fibroblasts, and found that the activity of one of the deiodinases that is expressed in the fibroblast, D2, was reduced. However, when we looked at the RNA, which is the gene transcription, it was normal. So that is very simple to interpret. It means there is no genetic defect in transcription, but in so-called post-transcription in the formation of the protein. And there are 2 possibilities. Either the protein is being degraded rapidly or is being synthesized slowly or less synthesized. So what happened at that point is what has become a little bit of a story in the lab. I had to take a trip, and when I came back, then my graduate students have found a mutation in SBP2. So the story is Refetoff leaves the lab and things happen. And maybe there's some truth to it. So, and the way she proceeded is she basically, because the family was big enough, did a study, so-called linkage or co-segregation, and found she could exclude all the known enzymes that are involved in degradation, and she moved in to the enzymes involved in synthesis, SBP2 being one, or products, not all enzymes involved in synthesis, and found that the 3 affected children were homozygous for markers in that gene, and therefore it was a good candidate, and found a mutation. As it happened, we had another family from Ireland that came to our attention only 3 months later. And would we have started with this family, we would have missed it, because in this family, they had 2 mutations. They were so-called compound heterozygous. They inherited a different mutation from the father and a different from the mother, the father being from Africa, and so not related at all, whereas the one from Saudi Arabia were related, were Bedouins. And so if we looked by co-segregation, looking if they were homozygous, we would have missed it because they have completely different mutations. But they had the same clinical phenotype, so it was logical, once finding one SBP defect, to pursue the others.
Have you generated an animal model for this?
So this is one of the projects I have. And we, at this point, we have already made the construct necessary for doing those mutations. And they are in stem cells ready to be injected in mouse eggs. One important issue there is that we know that if we completely knock out SBP2, the embryo doesn't survive. So the new mice that we are generating are a little bit more complex, in the sense that there are conditional knockouts, that we can turn the gene off at different times of mouse development, because we know that if it is turned off at conception, the mice won't develop. That will allow us to determine what selenoprotein is crucial for survival, that window that is necessary, and what kind of selenoprotein is required for that. So it goes a little bit beyond the thyroid.
In addition to the 3 syndromes that you've been working on, you're also working on thyroid hormone cell transporter, MCT8. What is the Allan-Herndon-Dudley syndrome? So this syndrome, first described by the 3 authors that carry the name,
was described in 1944 and was called X-linked mental retardation, and was within a large category of defects common in boys, and therefore they are inherited through the X chromosome, common in boys, that are associated with mental retardation. It turns out that in retrospect only, that particular family that was described has a mutation in the MCT8, but not all individuals with boys with mental retardation have it. And as a matter of fact, recent studies that have massively looked at boys with mental retardation, only a small proportion have this defect. What is typical to the MCT8 defect, and not in other cases of mental retardation, is that they have a so-called thyroid phenotype. It's always associated with the thyroid abnormality. And we believe that it's the thyroid abnormality that is causing the mental retardation. The thyroid abnormality is a high active hormone, T3, and a low inactive hormone, T4, and it is a bit of a surprise to have thyroid hormone deficiency when the hormone level, active hormone, is elevated. The defect is caused by the inability of this active hormone to penetrate the cell, especially the brain cells, and causing that thyroid hormone deficiency in the presence of high active thyroid hormone levels.
Are you using an animal model for this study?
Yes. So this has been actually the prototype of what I have mentioned earlier, of that process of identifying a condition, linking it to a genetic defect, and trying through the mouse to characterize it in better form and going back to the human. And in fact, this is one condition where through the mouse we've been able to identify a thyroid hormone analog that, despite of the transport defect, can enter the cell because it does not require that particular transporter. There are many other transporters. So this has been really our model for future investigation of other defects.
What analogs are you using?
It's an analog called DITPA, diiodothyropropionic acid. And it's been used in the mice and has corrected the thyroid defect. And it's not yet approved for use in humans in this country, but has been used outside the United States on compassionate basis, and has shown that it does correct the thyroid defects. Unfortunately, by the time the children that have received it were too old to correct the brain abnormality. This has to be implemented very, very early to be effective.
How early?
Probably before birth, because some of the damage is probably caused in utero, because the thyroid hormone that the mother provides cannot enter all the tissues of the embryo.
When you're working on your research, have you ever felt that you are in a race or a competition to get there first? Is that something that you've experienced?
Well, this is a very common phenomenon, I think, in scientific research. Sometimes these races turn out to be beneficial because it stimulates activity, and sometimes it causes disagreements, and there are many of them that we all know of. From my own point of view, I've had two instances of, actually three. The first one I just mentioned with Stephen Usala, which has been a very happy and very productive competition, which has left no scars. The other one happened later with the recognition of TTF1, thyroid transcription factor 1 mutation, which is also associated with a neurological defect, in which case Annette Gruters, who was the other laboratory, in her laboratory that this was recognized, we got in touch and agreed to publish our findings, which are identical in the same journal in JCI. That was not the case with MCT8, and if you permit me, I have some notes so I don't say something wrong. So the cases of MCT8 were brought, at least in my laboratory, to our attention by Knut Brockmann from Germany, a neurologist from Göttingen, on March 15, 2002, and in May of the same year from a physician from Canada, Thomas Best. And we got the samples from the laboratories in March 21st and July 3rd of 2002, at which point we started working on the deiodinases, as I mentioned, in this case also because of the discrepancy between the level of T4 and T3, in this case T4 being low, T3 being high. That was a possibility. And we didn't find anything in those deiodinases. So in September 8, I was in Gothenburg for the European Thyroid Association meeting, and I presented the cases to Joseph Curley. That's kind of interesting because he is a co-author in the other paper I'm going to discuss. So Joseph, who is a very nice individual, a good friend, I presented him the thing. He suggested that that there are two possibilities, that either the children had iodine deficiency, in which case the T3 can be high because they cannot add another iodine to the molecule, or that there may be involvement in the thyroid transport. Now, in the thyroid hormone transport. Now, I don't know where he got that, but certainly I should admit that that was what his suggestion among others. So in October of that year, Edith Frisma from Visser's lab presented the discovery of the MCT8 gene in the rat. And I was present, and I got up and I said, I wasn't sure. I believed it was a transporter because current dogma at the time was that the hormone is small molecule, it's lipid soluble, it just got into the cell by itself. And so I asked her, I told her, has you find a mutation in human to prove that this is effective? To which she said, she didn't give me a positive answer. So in June of 2003, I learned that Theo Visser reported a mutation in MCT8 gene in the Endocrine Society meeting in Philadelphia. So I was not present at the presentation, and next day I left for Italy, and I wrote him an email on July 11th telling him that I learned that they found a mutation and we have the same thing, and it would be nice if we published it in the same issue. So he said that was a good idea, but he had to consult with the co-authors. And I'm thinking in retrospect it makes sense because there are 17 authors in that paper eventually. So that was July 11, I contacted him. July 24th, he told me to wait. And on August 22nd, he said that it has been, I have to wait a little bit longer because there's vacation time and he hasn't had the answers. So on September 2nd, he sent me an email saying that that we should forget about it and that I should do whatever I wanted to do. And at that point, we sent our paper to Human Genetics. And it was published December 5th, electronically, and then in January 5th appeared in print. Now, their paper appeared later that year, I think was October of that year in Lancet. There was also another problem which was related to the editors of the journal. We elected to put a sentence saying that that the results will be presented at the American Thyroid Association, the results of VISER. Whereas because our paper was published later, they changed the tense and said that the results were presented. And that caused a little bit of a dispute.
Explain that again.
Okay. So in the original manuscript, we wrote that the result will be presented in the upcoming meeting of the American Thyroid Association. Now, the American Thyroid Association occurred after, as we wrote it, but when they published the paper, it was 5 months later, it had already occurred. So they changed the tense, saying that these results were presented. So Theo then had a long discussion with the editors. I was not involved other than I received the copies of that. And because were would imply that that we knew only after they were presented, whereas will be presented means that we knew before, which is true, and this is what we wrote. The tense was changed because the editor felt it already happened and the date has changed. So an erratum was published. It's a kind of an example of, I think, of the willingness that we all have to be the first and the willingness of recognition. And sometimes that becomes a little bit ridiculous. But anyway, that is my experience with that particular case.
Now, how do you feel about that, and how is there anything, what would you have done differently?
I think for me it's not very important. And when I publish on the subject, I always put our publication and theirs, and I don't put any dates or any precedent who published first or who published after. But I think for the young people it's very important because there is a young person associated with each one of those publications. And who has been, and the ability to recognize something de novo, for the first time, as compared to find the same finding that has been already found, is very different. And although it takes years to produce something, few months of difference may imply that the other person already knew about it. And I think that tense that was put there kind of reflects that point, which is a minimal thing that the editor felt that he was very accurate in describing whether it was will be or was. It makes a difference for the author saying, did he know before of this thing or not?
Is there more that you want to say on that?
No, I think if we come to as we become as older and recognized in our work, what is our mission in helping young people get into science and be successful? And we have to be cognizant of that. And these small things can be very hurtful for young people. There are other instances that have been debated, and I think the Endocrine Society may be able to do something about it. One is to protect the time of young people to do research when they are pressured, especially those in medical profession, when they are pressured to be involved in patient care. What can the Endocrine Society do is probably through the grants that they provide to young people have some influence on the institution to which the grants are given to guarantee that the time is protected, the time used under those grants is protected. The other thing is has to do with journals. Editors of journals want to achieve fame or want to improve the impact factor of the journal. The impact factor of journals is based on the number of quotations. And so it's one doesn't have to be that bright to conclude that if a publication covers a field that is very popular or is used by many investigators, the chances of the article being quoted multiple times is higher, irrespective of the importance of the article. So articles that deal with subjects with very narrow interest, even if they are very excellent, are being overlooked because that will reduce the impact of the journal. And I think that editors should make an effort to base their judgment and their acceptance more on the quality and the importance of the work rather than on its effect on the impact of the journal.
Speaking of the Endocrine Society, what has the society meant to you in terms of a community of scholars?
Well, the society has allowed me to interact with people that think differently, are in different areas of research, from whom I've greatly benefited. I think my research has always had a leap in progress when I encountered other people, and I think good examples are my sabbatical years where I was involved in work different than that in my laboratory. So coming to those meetings, I usually try to attend lectures that are not necessarily in my field in order to gain more knowledge, and especially those that have a technical importance in my work. The society has, I should admit that I have not contributed as much to the society as the society has given me. I've received several prizes from the society, but I haven't been involved in very much in the politics, and certainly not in any of the other administrative activities. And maybe there's a reason for that. I don't think that I'm that gifted in administration or in human relations, so the society probably recognizes that and keeps me away.
What are your current views of the field?
Of the field of the thyroid field? Well, the problem of the thyroid, and again, that's my field, and I certainly have some bias regarding my interest. I think the thyroid field is very interesting from the point of view of biochemistry, from the point of view of anatomy, and various aspects of that particular gland compared to others. But on the other hand, for many years, it has been recognized that giving thyroid hormone can cure the majority of conditions associated with thyroid deficiency. And therefore, it has not elicited as much interest from the government, from NIH, or from other bodies, because there is no need for very sophisticated drugs, very complicated treatments, like is the case with diabetes or other conditions. I think the few conditions I discussed that are unique, where thyroid hormone cannot solve the problem, are a little bit in the realm of those of diabetes, but there are few in between. And that's one reason that thyroid has not received thyroid research as generous funding like other areas of endocrinology. So I would like to see more recognition in thyroid research with respect of knowledge gain in basic sciences in understanding how hormones work rather than just centering on the necessity to do research in order to cure thyroid diseases.
Okay, thank you.
You're welcome.